This is a production of Cornell University.
Okay, here at the beginning, I'm just going to remind you guys again, my name is Bahia Zal. If you're a regular attendee, you probably already know that I'm a second-year PhD student in plant breeding and genetics program at Cornell, studying hemp with Dr. Larry Smart. Just so you know, these are going to be recorded. So if you want to share these with your friends, or if you want to watch them later, those will be available for you. Also, if you want more information, I will be posting the link to our hemp website here at Cornell. So with that, thank you all for joining us. And I'd like to welcome our speaker, Stefano Amaducci. Dr. Amaducci is a full professor of agronomy and field crops in the Department of Sustainable Crop Productions at the Catholic University of the Sacred Heart in Italy. He's also president of CityMap, a company that develops remote sensing technologies for precision agriculture. He has extensive experience on the agronomic evaluation of industrial crops for fiber and biomass production. And then more recently, he's shifted his focus to management strategies to increase sustainability in agricultural systems. To do this, he works at the intersection of crop ecophysiology, applied agronomy, and soil science. He also is working on agrivoltaic solutions to combine energy production and food production on the same land. He's coordinated the EU Multihemp and Hemps-sys projects and has participated in projects like Fibra, Grace, and SSUCHY, which are backed by the bio-based industries joint undertaking and which aim to develop high-value applications for fiber crops. Thank you, Stefano, for joining us today. I'll let you take it away if you can share your screen.
Thank you very much. Thank you very much for inviting me to this very interesting webinar series, and I'm very happy to contribute with my presentation, although I have to admit that when I was first contacted by Bahia, I was a bit hesitant on accepting, because even though I worked for many years on hemp, I have in the recent year drifted a little bit away from this crop. But then I thought it was a very good occasion to dust off some of my old work on hemp and share with you my long-term experience with this fascinating crop. So as you can see from the title, I will mainly focus on the effect of agronomic practices on hemp yield and quality and processing. I am an agronomist, as Bahia said, and my focus is ecophysiology of crops. So I think spent a lot of time doing this on hemp, but then of course I had to also work on other crops. And I think that this is, as you can see, for the occasion, I got from some old presentations some data, but also some images. And this is a picture of mine during my PhD experiments. I did my university degree in agricultural sciences, and I did my PhD at the University of Bologna. Here is August 1996. And actually, this was not the first year I was working with hemp, so almost 30 years ago. And I think it's interesting, and I prepared a presentation in which I will go through my experience, and I will show you my, let's say, personal hemp bio, as my career has been really depending, say, or founded on hemp. And I think it's interesting to consider that when I started work on hemp was really an abandoned crop. Actually, it was forbidden to grow hemp. So we had to get through a lot of problems, botherations, and permissions to be able to grow the crop. I was lucky I was not forced to follow completely the rules, because according to the rules, I would have had to fence my crop with 4-meter high fences and keep it illuminated during the night. Of course, these are regulations which are set in place for drug crops. There are plans, but I was, of course, didn't have the budget for such a facility, and especially we had to run also a lot of phenological experiment in which, of course, since hemp is a photoperiodic sensitive plant, if you illuminate during the night, you lose part of the sense of your experiments. I don't want to go too back in history, but I think I wanted to add also some of these pictures, because these pictures are present in the University of Bologna in our department, because hemp is part of the history of the agriculture in the region. Actually, I think it's shaped both economically but also physically the farms in the area. Physically, because you can still find the retting ponds, which are these basins in which farmers after harvest were putting the hemp to ret. And then there are still museums in which you can see all the tools and the machineries that in time were developed to do the processing. And actually, there is historical evidence that hemp was very important. There was even a death penalty in the case anyone would export from the town of Bologna any hemp material which was not before processed, particularly if you were exporting seeds. So there were some kind of a heavy form of protectionism. And this is also justified by the fact that it was very important for the economy of the region. And the main customer of the fiber of hemp in this region was the Royal Navy of UK. So you can imagine that in the 16th, 17th century, this was a very important customer. But this is just to say that despite all this history, the crop had completely disappeared in the 70s, totally, to the point that in 1975, when the law which rules drugs included cannabis, nobody complained by the fact that actually cannabis was an historically important crop, because nobody at that time was cultivating hemp, which completely exited the panorama of crops. But actually, my professor, Professor Venturi, who was the coordinator of my PhD, he started his research when he was a student on hemp in the 60s, and his research was on hemp because it was the last attempt, the last, let's say, project to try to save, let's say, this important crop. But as you see, this is I just put this picture. Actually, I could not find a more recent one. And from this, you might expect that after 2017, there was a big peak in hemp production, which was actually not the case. More or less, the acreage of hemp has been more or less stable or stagnating for the last year. But it was mainly to focus on the fact that when I started work on hemp, most of the hemp in Europe was grown in France, which is the only country actually in Europe that kept growing hemp, mostly for dual purpose crop, which is making paper pulp, and then harvest also the seeds for different, especially feed applications. So there was not much knowledge. It was quite difficult, and so actually the first project in which I worked, so this is my hemp peer, it was in 96, 98, in the frame of which I did research which were pertinent to my PhD. It was called Hemp, the acronym of Hemp for Europe Manufacturing Production System, for the project, a European project, which tried to really collect the few researchers or few also companies that throughout Europe were interested in hemp cultivation. And in a certain way, there was a part of the project, of course, which was focused on agronomy, because most of the people, let's say, forgot how to cultivate hemp, but particularly nothing was done on hemp in the last 20, 30 years. So let's say the agronomic technique had to be at least revised. there was practically no mechanization, but also there was also a big lack of knowledge on the quality of hemp and its suitability for different production system, for different end-use application. Just to give you an idea, it was coordinated by UK. There was about 1,000 hectares of hemp grown in UK, and it was grown for horse litter. So the main product was actually the byproduct of hemp, so the woody core, and they were looking for a potential application for the fiber. Just some results. Of course, I have a kind of a broad literature of work I did in collaboration with different institutions throughout Europe, and I will not very much focus on numbers and figures and tables, otherwise it would take too long, but just let's say on main results or which were the objectives, let's say, of these trials. For example, the first set of trials which we carried out in the frame of this hemp project were carried out in 3 locations: UK, Italy, and also the Netherlands. The Netherlands was involved because in the University of Wageningen there had been a large project to promote hemp in the previous year, which also financed the research of 2 PhD students. I think that many of you who work on hemp know their work, which was published in 1994, more or less. And then we continued this research, mainly try to focus on 3 agronomic aspects, which is planting density, the effect of variety, and nitrogen fertilization. So, of course, as you can see from this graph, and we made this publication, which is still one of the most cited publication, or the most cited publication I have in my scopus, also because I think a lot of people have been citing this publication in the introduction on their paper on hemp. And but it was interesting because it shows which is the effect of planting density, variety, nitrogen fertilization on the main traits, of course, dry matter yield, but also how the density, for example, as you can see in this picture, affect the way of each single plant. Of course, the denser is the crop, the smaller is the plant. And we were very interested in this aspect because at the time we didn't have many techniques to evaluate the quality of the fiber and also the amount of fiber produced by the plant. So we made some calculation, like here you can see average cortical surface per single plant, which of course on a unit area increases with planting density. So the hypothesis was that planting more plants per meter, we would have more fiber of better quality. Then I also focused my PhD on some ecophysiological studies. So different densities. So I went with my septometer to measure the light interception. So how much light was absorbed by the plant with different density. I calculated the coefficient of extinctions. So I started to collect the data, which at the time were not available on hemp, but also especially always focusing on the effect on yield and on plant height and stem diameter. And also here we started to develop the idea that it was important to have thin stems because that would affect not only fiber content, but also the processability of the plant itself. So the effectiveness with which we could extract fiber from the stem. But as I said at the time, we didn't have, say, the machineries or even the industry that could be involved to do this trial. So after the end of this project, I go back to my personal bio. Actually, the partnership of hemp split in 2, and one group, which was more, let's say, composed of geneticists, went and wrote a project called Harmonia, in which the idea was quite interesting because they were working on a yellow variety. there are naturally, or at least they were already present and commercially available, some yellow varieties. And by yellow variety, I mean really that they have a yellow stem and also quite yellow leaves, with the hypothesis that these yellow varieties could be processed, so the fiber could be extracted without retting, because actually retting was one of the key points that rendered very difficult, if not impossible, fiber extraction compared to what it used to be in the past, according to the tradition. The other group, and I was part of this group, and I also wrote and coordinated the Hemps-sys project, which was financed 2 years later compared to Harmonia. And the focus and the idea was to study a production chain. So we really started to develop the idea that hemp could be grown not just because it was a fantastic crop or wonder crop, as many people were believing, but because you had a complete production chain in which, of course, adapted suitable varieties were commercially available. There was an agrotechnic which could be, of course, adapted to the environment and to the end-use application. The mechanization would be in place, and of course there would be both the processing facilities to extract the fiber and a market for the fiber. And the idea here on Hemps-sys was to target high-value application, and particularly textile destination. So the idea was that with textile, we could have achieved a price for the fiber, which could have easily supported all the cultivation and the rest of the production chain. And we had among the partners Linificio Canapificio, which is a historical company that in the last years has worked mainly on linen, flax for fiber, but in the past, as the name says, because it's Linificio and Canapificio, Canapificio is a hemp industry, flax and hemp industry, was also in the name and was very important in the past. So we brought a project in which there were like different, work package, and each work package was representative of a phase of the production line. So there was the production of raw material with agronomic trials, processing of the raw materials after harvesting, the transformation, the extraction, the retting, and then the production of yarns, and finally also some design and application consideration, also a marketing studies. And then there were some transversal work package, which were quite important because what we were lacking in previous research was the capacity to characterize the quality, and the quality had to be characterized throughout the whole production chain. So to know which was the effect on end-use application of specific variety, density, processing technique, and so on. One innovation that we brought in the project at this level was to reverse what was the, let's say, the traditional processing line. So after cultivation and harvesting, traditionally we had retting. So after retting, we had the industrial processing, which is a sequence of scatching, decortication, scratching, softening, hackling, spinning, weaving, and finally the production of textile apparels. In our project, instead, we reverse this phase in the sense that we didn't have the retting before the decortication, before the scratching, but we had the scratching on, let's say, what we call green material, and the degamming or the retting would have been done industrially later on the decorticated fiber. This would have rendered possible to achieve a good quality without doing retting, which was unfeasible, even forbidden. You could not, use a natural pond to put water, or rivers as we have done in the past. The environmental condition which render possible field retting or dew retting, which is done normally in the north of Europe with flax, was considered not suitable, the environmental condition in Italy, for example. So this would have, let's say, avoided all the environmental problems, which are anyway present also with, of course, lean and flax production, but for sure we would have had in Italy with hemp. So this was the idea. Okay, we had we kind of broadened this the spectrum of conditions which we had in the previous research. So we also have Finland and Sicily, besides also the Netherlands and Hungary, and we address more factors, agronomic factors. So harvest time, stem portion, plant density, water availability, especially in Sicily, genotype again, comparing monoecious with dioecious variety, and also the environment, so different environment and different years. The plant portion is because we knew that there is already the problem of homogeneity is quite large in hemp. There are usually over 100 plants per square meter. The genotypes are not hybrid, are not really homogeneous, so there is this big variability within your crop. And also we notice, of course, that the base of the plant has a lot of secondary fiber. The fiber are coarser, more mature compared to the top of the plant. So the idea was to set up and first characterize this diversity along the stem. On the other side, it was to cut the stem in two portions and keep them separated, because in this way we thought we have two different quality, we keep them separated, and then if we want we can mix it at industrial level later, but at least we increase the homogeneity which is required by the industry. So again, we didn't have a lot of methods to measure fiber thickness, fiber maturity. So we did a lot of work. I remember the PhD student who did this work. I mean, it was quite tough. And we were measuring fiber diameter, fiber maturity index, the width of the bark, width of primary fiber, number of cells of primary and secondary fibers. And when we wrote a bunch of papers in which we showed that the fiber diameter, so the fineness of the fiber was the fineness was increasing, so the diameter was decreasing with plant density. So the higher the density, the finer were the fibers. At the bottom of the plant, of course, the fiber was coarse, but also especially was more mature, because the maturity, we find a nice evolution independently of the number of nodes with time. So we kind of not validated our hypothesis, because our hypothesis was that to keep the two parts separated, we would have a better control on quality. But we confirmed the fact that fine fiber were on top and less mature, but probably also less mature meant also of better quality for some textile applications. But then we had unexpected results when finally we could gather enough material to arrive to do an industrial processing with our fiber, which was a lot of work because there was not proper mechanization. So, I mean, I spare you all the trouble we went through to have 100 kilos of fiber to get into the industry, but we had unexpected results. So first of all, the highest fiber yield after scratching was in the second harvesting and at the bottom portion. So second harvesting was okay. Second harvest was at full flowering, which was not particularly unexpected. But the fact that we had the highest fiber yield here, you can see here in the bottom portion compared to the top portion was a little bit unexpected. And one of the explanations we had, because we expected the bottom portion to be more lignified, to have more secondary fiber, so to have more difficulties to separate the fiber. But actually, it was for the fact, you can see here, that the number of shields, the hurts, the little bit of wood that remained attached to the bast fiber were dependent on the diameter of the stem. So they were higher at higher density, but also on the top of the part, which has a lower diameter. So more sheaves attached during the phase of scratching when you try to clean out the sheaves. of course, the impact determined these fiber losses. So these were some unexpected results, which anyway, unexpected result still is a result. And then we had anyway some conclusion from the industrial trial. So in agreement with previous finding, the optimal soil density was about 120 plant per square meter, and the best harvesting was at full flowering. The capacity we had to control plant height, which was quite important because actually we had to make the hemp stem fit into flax machineries because there were no hemp long fiber to make textile of high quality textile on the full stem. So we have to cut in two fractions, and so had to be more or less 2 meters. And so we could control the height of the plant through planting density, harvesting time, and also the genotype, which control phenology. So the height of the plant at flowering is determined until the end. Then we concluded that due to the limited quality variation between the two stem fraction and also the unexpected result we arrived, it was not justified logistic effort necessary to keep the two stems separated. There were other interesting results though, and further conclusions. And we involved some nice designer from Bologna who managed to produce some cool objects and apparels with the fiber we produced at industrial level. And they also did a marketing study. And the outcome of this marketing study is that to support the hemp production chain, we had to have high added value products. And another thing is that the marketing strategy should not be on the typical narrative that hemp is the crop that will save the world, and with typical, leaves, which maybe reminds about some other application of hemp. So the idea was, no, you have to, and they created this logo, blue hemp, blue as the noble principle, and some green in it because it's environmentally friendly. But this was the narrative that had to be pushed forward in order to support high, so expensive product from hemp. And then, of course, to work on mechanization and fiber separation because the machine were not appropriate for hemp, especially the harvesting. Next project is Multihemp. Multihemp came, as you see, some years later. I didn't stop working on hemp during this period, but actually I was told that if I wanted to continue my career and become a professor, there was no place for hemp professors, so I had to work also with other crops, and so I did. But when there was this call from the European Commission, which was asking for multi-purpose crops for industrial byproduct and biomass to support the bioeconomy, the bio-based economy, then it seemed to me that this call was written for hemp. And in fact, we wrote a project which was called Multipurpose Hemp for Industrial Byproduct and Biomass, and we were financed. We were first. It was a big success, and the idea was really to promote hemp as a natural biorefinery, which is the case because you all know that hemp is not just fiber and wood, but it's also seeds and it's also flowers, cannabinoids. So there is a lot of properties and products in this crop which actually have a real benefit, have a real market. So we did this poster during the Multihemp project, which was also in parallel with another European project called Fibra, and in which you cannot read very well, but you can download it is this poster. These are real data about how many tons of these different products are on the market. So why to promote hemp for the bioeconomy? And actually, the idea was hemp could be a model for the bioeconomy. So the bioeconomy, the bio-based economy, especially in these years, in 2010, 2012, it was just at its beginning. So it needed to be promoted for people to understand what it was. And I thought is that it was a perfect crop to promote bioeconomy, because it's really a crop which you can use every part of the plant in a complete value chain. It produces also food, which was another important issue for a biomass crop, because in 2008, you remember there was this big problem of the food versus fuel debate, which has not been solved as far as I can tell so far, but still hemp produces also food and feed. So it's, let's say, perfect also in this sense. Then it's a low input crop, high resource use efficiency, and we have to prove, of course, all these things, but it was already partly demonstrated. It had additional environmental benefit. For example, it can be produced on marginal per unit land, and then, of course, it produces innovative, recyclable, sustainable material that have a market. So that was a good idea. It was a large project, a lot of partners, and not only we consider suitable hemp for the bio-based economy, because it's a natural biorefinery, but we also want to build a biorefinery concept for the crop. So we started to implement some innovations, and this time we compared the longitudinal hemp, so it's the same one that we tested in Hemps-sys, which is currently used in linen and fiber flax, or the disordered hemp, which is most of the hemp processing facilities do not keep the stem parallel, but you just process the whole stem. But the idea was to harvest hemp, okay, in different moments, but the idea to make it a multi-purpose crop, to harvest a seed maturity, to use the seed, of course, but also to keep the threshing residue. So, of course, the threshing residue are all these bit of leaflets, which surround the seeds, which were the same leaflet which surrounded the flower and which are rich in cannabinoids. So the idea was to extract cannabinoids, so to have like a multi-purpose crop, not just a dual purpose, but a triple purpose at least. And then to study, of course, all the production chain until the end use application. There was a lot of work in this project. I've really no time to go through any of it, especially because there was a lot of research done on genetics. So we did reverse genetic association mapping, and it was a good excuse to do that, because so far none of these molecular technique had been used on hemp. So we had big trials with more than 100 varieties that were sequenced, and there were nice work on that and nice publication out of that. But on the agronomic side, which was the one I was directly in charge, we did again multi-location trials, and then we also developed modeling, both modeling phenology, which actually we did already in the frame of Hemps-sys, but also we work on photosynthesis, on crop growth, and the effect of nitrogen again and planting density. Hemp phenology, I got a bit fast. I think is one of the most striking effect you can find. So this is the same genotype grown in Latvia, which was a partner of the project, and the same grown in Italy. So this is Finola, which is, you might know, is one actually of, I don't know if it still is, one of the most widespread variety, because it's grown on, or used to be grown on more than 10,000 acres in Canada. It's really derived from some land races in North Russia. So it can flower also with very long days, because as you know, hemp is a short day plant. So it flowers when the days are short. If you go to the north, the days are very long. So in Italy, days are short and the plant flowers immediately. So we developed this crop model, which I don't have time to show, but there are publications in which is described, and it works quite well. You see, this predicts very well, for example, a problem that we had with certain varieties, and I think is a problem that you know very well in the United States, because there are very few varieties, or there were at least, that were developed in the United States, and most, at least in Europe, are developed in France. So if you go from France to Italy, you go from north to the south, and then most of the varieties that were bred for France were not really suitable. And in fact, if you were planting them at the right time, for example, end of March, beginning of April, you had some pre-flowering because the day was too short. And the model works quite fine, so you can make also simulation study to decide which is the best variety for your environment, provided, of course, that you have a calibrated model. And this is the model calibrated on all the environments involved in the Hemps-sys project. Then we have also here a bunch of results. And as you can see, the phenology is important because the longer is vegetative phase, the higher is the biomass yield. But on the other side, the lower is the seed yield. Seed yield, I will say something later because it's quite complex, but these were the results, of course, of our study. We also worked on photosynthesis, both on plant photosynthesis and crop photosynthesis. We make nice paper. We calibrated a crop growth model, but we also worked on a nice comprehensive study on planting density and nitrogen fertilization. I wanted this to be not the last study, of course, but at least we found that no significant interaction was found in between density and nitrogen. So since it's quite expensive to do field trials to study interaction, I think it was a nice result. No longer do like that density nitrogen interaction because you don't find it. You don't find it positive. Then we found that the best planting density for fiber production between 90 and 200, for seed production in between 30 and 75, and for dual purpose in between 90 and 150. Nice results on nitrogen. These are many studies we did, and above 60 kilograms of nitrogen per hectare in most of conditions. We didn't find any response on yield. We also did some nice dilution curves thanks to our experiment, and hemp performs incredibly well. You see the minimum nitrogen content or the critical nitrogen content. So the critical nitrogen content is the percentage of nitrogen in the plant under which you start to have a reduction in yield. Is quite low, and this is due to the fact that actually hemp has a very good way to recycle nitrogen. So it accumulates in the biomass mostly carbon. There is very little protein in the stem, and the nitrogen recycled from leaves to leaves. Okay, and things change, of course, after flowering. But the determination of hemp seed production, it's always been a very difficult task. Actually, hemp has not really been bred like many crops we know for seeds to produce seeds, or at least there is a big amount of seeds which is lost. There is problem of shattering. The homogeneity of seed maturities, it's very low. Then when you study, for example, quality of seed, actually you have a lot of cross-pollination, so you're not even sure that the oil content from one variety is really of that variety, has not been contaminated or cross-pollinated by the rest. Then the birds love hemp, so you should protect your crop from birds, otherwise you lose a lot of seeds. This is just an image that Kyle, one of my PhD students, took from the same inflorescence. You can see you have little green seeds and mature seeds. So you have to take, a choice at a certain point. You want to make these seeds ripen, but then you lose those ones. So it's very difficult to assess the seed yield in hemp, at least at plot level. Then we did some characterization of oil content. But also very interesting was the THC concentration of the different varieties and the CBD concentration, because in the meanwhile, it was during the year during Multihemp, there was this big hype on CBD, and actually our idea was ahead of this big booming interest, and we were, as I said in the beginning, the idea was to collect the threshing residues. And we made this calculation. It was the amount of kilograms of CBD that we could have harvested or collected with the threshing residue. So for sure, this was an interesting opportunity to gain, value along the hemp production chain. The last step, the last years after Multihemp, I was involved in two other projects, Grace and SSUCHY. I would just say something about Grace. Grace actually was a very big project that was meant to find industrial crops to be grown on marginal land. And honestly, I felt it was a kind of a pity to relegate hemp on marginal land. My intent, okay, besides, of course, research targets, which are the research targets more or less I try to show you during my presentation, I would have, of course, liked to see hemp being cultivated industrially, at least in the Po Valley where I live, or in many other places. And I believe that the hemp has a very interesting place in a crop rotation. There are many evidences that the crop which follows hemp benefits from the presence of hemp, which kills the weeds, leave the soil in very good conditions. Then it produces also food, so why to relegate hemp on marginal environment? But of course, okay, I anyway included hemp in this project, and we did some trials which were anyway interesting, because we tried to grow hemp on mountain area on the Apennines, where of course we consider this as marginal environment. We did genotype screening for osmotic stress, heavy metals, and also a review on how to adapt hemp cultivation to marginal environment. It was good to see that there is a certain variability in the hemp genotypes that we try for osmotic tolerance, for zinc and copper. So we did a paper with this ranking, and also we did this review in which we find that it's very difficult, because the idea was to consider hemp as a multi-purpose crop, but it's difficult to keep hemp as a multi-purpose crop. For sure, if you grow hemp on contaminated land, you cannot think about using the seeds for food and feed, or to use the cannabinoids for pharmaceutical application. And the only possibility, and actually it seemed quite interesting, is to grow hemp as a dual-purpose crop in mountain area, where, for example, you can grow it organically, and you can get nice added value from the seeds and from the oil. Another big chunk, I would say, of our research was on this yellow variety. Many yellow variety, not many, but few yellow variety appeared on the market, and we did a lot of studies, and in fact we proved that they're easier to decorticate, so they can be a good solution when you have difficulties to extract your fiber, because retting, for example, field retting is not feasible, and water retting, I don't think it's really a solution for it. I would like to conclude with the outcome of stakeholder meeting we held during one of our meeting, or I think it was in occasion of the annual meeting of the European Industrial Hemp Association, in which we had the processor, farmers, all the stakeholder, I would say. And then I asked these two questions: what are the main technical constraints of the hemp value chains, and what is needed to increase hemp cultivation in Europe to 100,000 hectares? Okay, and this is more or less the answers, which I think could be a good point of discussion, also a way forward. The first is to enlarge the market of hemp products, because it seems that there is a huge market for hemp fiber. There is so many applications, but then if you ask the processors, or at least this was until 4 or 5 years ago, most of the processors that we have in Europe, they can process up to 2,000, 3,000, 4,000 hectares, but most of them just grow 1,000 hectares, because 1 year they cannot sell the fiber or the sheaves. There is not a stable market. So this makes me think that maybe they said this. So you have to enlarge the market, and you have many ways to enlarge the market, of course, to find mainly products that have higher properties, higher qualities, and promote them and make it a commodity. Then you have fiber quality issues, mainly weather dependent. So cultivar ability to get out from the field the crop as soon as you can it's a good way. And then also maybe develop a due rating model. So to try to simulate, and this is something that actually we are still doing, to try to combine which is the best variety for a specific environment in order to have, you know, enough time for the ratting and the right condition before, of course, the next crop. It happens sometimes that the conditions are not good, and then you have to leave the hemp on the field the whole winter. And then, of course, you can skip a crop. This is not acceptable for many farmers. Then still there is a grading system missing, so it's difficult to, you know, to communicate in between processors and industries. Mechanization of harvest. I mean, this is a big topic, and I think a lot have been solved in this sense. Another important point, I think, is long-term cultivation, so stay by contracts. If you have to invest in a company, you want to be sure that farmers will keep growing hemp. I have a lot of anecdotes about this, but I think I finished my time. Another important point is the size of the processing plant. I think that it would be ideal to have a small processing plant which can be managed by just a few farmers. But it seems, at least in Europe, that the size of the industry is around these 1,000 hectares, which is still a good size, manageable by large group of farmers or cooperatives, and it worked quite well. But I think it would be interesting to have also a solution for more artisanal, let's say, application, which would, I think, help to have hemp penetrate more into the market, into agriculture. And of course, new varieties improved for the quality and for the seeds, especially with a good level of cannabinoids. There is also a lot of work to be done there. So I conclude with this picture where you can find me. There is my address, but there is also a picture of an agrivoltaic because yeah, most of my time now, this is my next, let's say, niche application. I was also a pioneer working 10 years ago on this, and okay, it's giving me a lot of satisfaction as hemp did in the past. Thank you for your attention, and I'm ready to answer your questions. Sarah asks, how do Europeans use language to distinguish different parts of the hemp plant? Fiber as the whole stalk and not just bast fiber. Do you think this language should be changed? There was, I think, a lot of discussion in the past. It's not only, I think, Europeans, but it's within Europe. I would say that if you talk to if you have a complete group of stakeholders, they mean by fiber something else. We were with botanists, and for them the fiber was the single cell. For other was the bundle of fibers. For sure, when you talk to the industry, the fiber is bundles of fibers. I don't know. I think we could find an agreement when we talk about stalk or stem. I think that we usually use. I mean, personally, I always use like bast fiber to define the long fiber, and sheaves or herds to talk about the wood, which, if you want, from a botanical point of view, they could still be defined as fiber. So I don't think there is a recognized and agreed upon way to define fiber. But when I said fiber, I really meant the bast fiber used for textile or technical applications. Wayne, hello. I work in hemp fiber USDA and follow your previous hemp work closely. How did you identify hemp fiber properties such as fineness, diameter, and length in your study? In general, what instrument technique did you use? Okay, this is I think I showed in my presentation that the first time we tackled the problem of fiber diameter, we were measuring, as you saw, on stem cross sections. Actually, the stem cross section, the technique was botany professor from the University of Parma, which is present in one of our papers from 2005. It was really to cut with a blade, a razor blade, thin slices of hemp just harvested, so fresh. And then we were coloring it, and then under the microscope we were taking pictures, and then the picture was stored, and then during the winter we were measuring the diameter of each single cell. And then we were measuring external, internal, so we were measuring also the lumen, so the degree of maturity. Then we did a nice work with Professor Musik, and he's one of the colleagues that becomes a dear friend. We had a long time collaboration, and he always worked from the technical side. So already in Hemps-sys, but also in Multihemp, I didn't go through that, also because, let's say, not the core of my competence, but we developed a set of machineries. So I also have in my lab a small decodicator. So we measure decodication, decodicability. So how many times you have to pass through the machine to get rid of the shifts, and in the end, also after 6 passages, how many shifts you have. And then the basts were cleaned, and this, of course, is just the bast fiber, was opened with a core separator, which is another experimental machine, which I do not have, but Professor Musik has, which is kind of a card which opens the fiber. At this point, he was measuring fiber fineness, both with the Shirley method, but also with the fiber shape. And I think in 2018, we also made a publication in which we compare the results of Shirley methods and the fiber shape, and also the measuring of each single fiber, which we were doing on the samples, and there is a good fitting. So this is a way, of course, to measure. Otherwise, and this was always a big problem for us, we would have to produce a lot of fiber, process it through the industrial machine, and then ask Linificio, which has always been our industrial partner for the characterization of the fiber for textile application. And then, of course, they have their own method with airflow and other system to measure fineness. But of course, we wanted to link the fineness to agronomic parameters in small plots and with replicates. So in their case, we needed kilos of fibers. For us, was impossible to do. Are there any application of hemp seed hulls post-oil extraction? Well, I don't know. I heard, of course, that there are different uses. I think the most common application is to use as a feed. And actually, still now, I think that the biggest application of hemp seed is for feed, especially in birds. So you can extract oil and still use it for feed, but has been used also for a bunch of food application. For example, making, I saw pasta made out of this flour after oil extraction, or making biscuits. Of course, in different proportions, but I don't know if there is a more precise commercial application. can seed lose viability if they're not harvested within the proper time frame? Good point. As I said, seeds, I'm a bit ashamed about this because I've done, I don't say 30 years, let's say over 20 years of research in the field on hemp. But seeds have always been the weak point. Okay, first of all, I mostly focus on fiber, and seeds. The few times I approached seeds, it was always complex. Already hemp is complex. You have a lot of plants to count. There is a lot of variability and a lot of problems along the way. And to collect seeds has always been a challenge. But I can tell that there are a lot of problems linked to seed quality for germination. And I can say, because we collaborated many times to try to maintain also the traditional varieties. But for sure, the French have a good answer to that. I visited them many times, and they have a very good organization on when is the right moment to harvest. And I don't think it's a problem of viability. It's a problem of having your seeds falling on the ground. And in fact, anyway, when you harvest seeds, also for multiplication purpose, and you plow, you leave the soil, you can see like a carpet of small plantlets, because it's really a lot of seeds are lost. So I don't think the problem is really losing viability because you harvest too late. But if you harvest too late, you lose your crop. Actually, what is very important is that you immediately dry at very low temperature the seeds to keep its high germinability, and also to clean the seed, to sieve with densimetric and all these machine, which I don't know the name in English, I'm sorry, to clean the seed, to prepare it before storage. So they also eliminate small seeds, big seeds. So what is left is average seeds dried immediately after harvested, or within really 12 hours, in order to prevent problems in the seed. And also how you regulate your combined harvested, because if you're too impactful, you can easily damage the seed. That is how they reach this fantastic germinability of over 95%, which is, in my experience, unprecedented. In one of your paper, you mentioned the relevance of lumen area to cell area of primary fiber to determine maturity index. What is the importance of this parameter for textile? Do we want higher or lower maturity index? This is a very good question. We found in literature that a low maturity index, so let's say a high maturity is when the lumen it's filled up. So a low maturity should be connected to fibers more flexible, let's say, with a better tensile strength. So this was the idea. So we measured in this sense, but we never had a chance to test these fibers that we characterize for tensile properties, for example. So I cannot answer. I was not able to validate these hypotheses, which I found in a paper. Hi, Andrea. It's good you're still interested in hemp. There was a comment from Andrea Herman. Can agrivoltaic be used for hemp? I hope so. For sure, I will. Actually, I already did. Because of this picture I showed you. It's an agrivoltaic plant that has been up and running since 2010. So for over 14 years. And 1 year I tried hemp. It was not particularly successful, but not because of the agrivoltaic. And actually my idea, and we also tried with linen, is the fact that with shade, as with density, the plants grow taller and thinner, and in a certain way, you could stretch the fiber a little bit more and get a longer fiber. This is again an hypothesis that I'm looking forward to validate. Biotic stress from Smart Lab. I've seen some relationship between them and fiber quality. No, I didn't. Well, I had my trouble to study relationship in between fiber quality and planting density and harvesting time and variety and also nitrogen content. Nitrogen content was also some challenge because also there, and somehow not really fiber quality, but processability of the plant. Yes, there was this idea, and it was in this empirical French literature, so from experience, that if you give too much nitrogen, then the stems are more difficult to decorticate. It would be nice to know why from a biochemical point of view. And we also saw this with our decortication, and we have a paper out in which we show that with higher nitrogen level, the decorticability decreases. But we were not able to link, the biotic stress and quality. Laura is processing fiber for research on an antique flux breaker. I work for University, Hemp, let's work together. Yeah, it's cool. We did the same actually in Hemps-sys. There are a few papers out in which the hemp was water retted in Wageningen actually, and then it was scratched with a Flemish mill, which I guess is something very similar to what you are using in your experiment. Okay, I think questions are finished.
Thank you so much. That was great. It's great to hear about some of the work going on in Europe that hopefully the US can learn from as we build up our hemp industry here. Our last session, as I mentioned, will be in 2 weeks with Shelby Ellison from the University of Wisconsin-Madison. She'll be talking about genetic diversity of US feral hemp. So thank you again for Stefano for joining us today, and I'll let you all enjoy the rest of your day. Goodbye.
Thank you to you. Thank you to you all. Good luck with your research. Bye.
This has been a production of Cornell University, on the web at cornell.edu.